The history of bioinformatics is very old, way back in 1865, when a monk named Mendel has performed some experiment on pea plant, and he has come up with some very interesting laws of inheritance. So with the passage of time, we will see many more discoveries leading to the field of bioinformatics. But in this brief history of bioinformatics, we cannot cover all the historical events that lead to the formation of this very important field. But we have selected some of the historical events. So the events that are not included in this history doesn't mean that they were not important, but only selected events they are added in this short history. I think it is much better that before going to the history of bioinformatics, we must know the basic definition of bioinformatics. So bioinformatics is an interdisciplinary field that develops method and software tools for understanding biological data. As an interdisciplinary field of science, bioinformatics combines computer science, physics, mathematics, medicine, molecular biology, and many other fields to study and process biological data. So as I have told that the history of bioinformatics starts way back in 1865, when a monk named Georg Mendel, he worked on pea plants and come up with a very interesting laws of inheritance. So Mendel has tracked that segregation of parental genes and their appearance in the offspring as dominant or recessive traits. And he has presented three laws of inheritance. One is the law of segregation, law of dominance, and law of independent assortment. Next is Thomas Morgan, who in 1910 performed some crossing over experiments by using an experimental fly known as Drosophila melanogaster. So his main finding is that he has proved that genes responsible for the appearance of a specific phenotype were located on the chromosome. So this is a very important discovery that is made by Thomas Morgan in 1910. Then we can see in 1951, two scientists named as Linus Pauling and Robert Corey, they have for the first time predicted the structure of a protein in the form of alpha helix and beta sheets. So they have predicted very important structure for the proteins. Next is a very famous two scientists, Watson and Crick. And we all know that in 1953 they have proposed a very famous double helix model of DNA. And on this discovery they were blessed with the Nobel Prize. Then came the Frederick Sanger, who has for the first time sequenced the bovine insulin. So we can see here that this is a very important discovery. So he sequenced all of the amino acids present in the bovine insulin. So the origin of bioinformatics can be traced within the sequences of proteins and DNA. So here we can see a lady named Margaret Dayoff in 1965. She has worked on the sequences of the proteins that were available at that time. And we can see that most of the databases, and one of the most important database, UniProt, that we use every day is based on the work of this lady, Margaret Dayoff. So I think that most important work that lead to the formation of the field of bioinformatics is based on one of the works of this lady in 1965. And she has also written a book named The Atlas of Protein Sequences and Structure in 1965. So she has also compiled a book comprising of all the sequences available at that time. So in 1970, a scientist named as Pauline Hogweg used the term bioinformatics for the first time when she was using the conversation with her colleagues. So this is again a very important event in 1970 that the term bioinformatics was used for the first time. So here we can see that in 1972, Paul Berg was successful in creating a recombinant DNA techniques and lead to the formation of recombinant DNA. So in this technique, he has used some foreign DNA and then clone it into some plasmid, and then plasmid is then transformed into some bacteria, and that foreign gene was expressed in that bacteria. So again, it is a very important discovery, and this recombinant DNA techniques, they are being used today very frequently. So again, next, a very important event took place in 1977 when the first organism named as bacteriophage PX174 genome was sequenced. So its whole genome was sequenced, and that was the really a great discovery in 1977. In 1977 again, Tom Coates, he has worked and established a database that we all know today is known as Protein Data Bank. In Protein Data Bank, all of the structures of the protein that are discovered today are present in this database. So 1977, the year 1977 is very important. As we see, the first organism was sequenced, and again we see that in 1977 a very beautiful method was proposed by Alan Maxim, Walter Gilbert, and Frederick Sanger, and in this they have proposed the sequencing method. Alan Maxim and Walter, they proposed the chemical method, and Frederick Sanger, he has proposed the biological method, how to sequence an unknown gene. So the Frederick Sanger method, biological method, is still being used today with some modifications. The biological method proposed by Sanger in 1977 is being used commonly today. Most of the labs, they are working on recombinant DNA technology, and by using these recombinant DNA technology, they have to sequence the unknown genes, and for this purpose they have to use the Sanger method proposed by Sanger in 1977.